All right. Excellent. So let's see, I will eliminate this so I don't have to look at this and we have a bigger screen.
Is that everything? No, that's
No.
no, you don't want
No,
to do
you don't
that.
want to do that. No, you don't want to do that. Okay. Click somewhere else. Okay. All right. It's 7 o'clock, folks. We're going to get started. Good evening and welcome. I'm Mila Stroganoff here tonight as your host and facilitator. The board of the Friends of San Pedro Valley Park, Adrian, aka Mr. Zoom, and I, Mrs. Zoom, of course, wish all of you a joyous holiday season. Merry Christmas, and may your new year 2022 be filled with good health, happiness, new joys, and new adventures. I'm pleased to announce that the Friends will be continuing the lecture series in webinar form while the visitor center is closed. So we have a series of lectures coming up in 2022. I've scheduled Zara McDonald, researcher and founder of Felidae Conservation Fund. And you know her well from the lecture that she gave us in 2020 in August on the mountain lions. And because of the fact that we had so many wandering around, we asked her to come and she spoke to us then. She'll be with us and she will be joined by Alice Granados, who is an ecologist at Felidae as well. The date, please mark your calendars. The date is Tuesday, January 25th at 7 p.m. And the subject is our beloved bobcat. The flagship of our organization in the park. So bobcats is the title. The title of the webinar is Bobcats in the San Francisco Bay Area: Biology and Ecology of a Native Mesocarnivore. So do join us. As an aside, as an individual that even though I'm very much a part of the Friends and the board, I just wish to bring your attention to the fact that there is a major construction project, housing project that's going up or planning to be built on San Pedro Mountain in Lindemar area. And it's not just the concern of those who are here, but it should be the concern of everyone. And if you go on the website savesanpedromountain.org, you will find lots of information there. Besides the flora and the fauna, the topography changes, retaining walls are built. Nothing is quite the same after you have a huge housing project built. So if you are even minimally interested in not being inundated with a huge housing project, please pay attention to this event that may be around the corner. That's all I'm going to say about it, but it's definitely something we should take into account. And we have with us, I'm very happy to say, we have Dr. Alejandro Velez Melendez with us. He's very kindly agreed to join us and give us a lecture on the ribbit of Pacific chorus frogs. There's more than meets the ear. Now, you've read all about what he's going to talk about, but I will give you the bio and introduce him. Alejandro joined San Francisco State University as an assistant professor of biology in 2017. He holds a Bachelor of Science in Biology and a Master of Science in Physiology and Behavior from the Universidad de los Andes in Bogota, Colombia. Alejandro obtained his PhD in Ecology, Evolution, and Behavior from the University of Minnesota and conducted postdoctoral work on sensory ecology at Purdue University and on evolutionary neuroscience at Washington University in St. Louis. In addition to his work on North American frogs, he has studied communication and sensory perception in songbirds, electric fishes, and tropical poison dart frogs. Whoa, yes! Alejandro is also a scuba dive instructor and enjoys exploring California's natural wonders. So I shall turn this over to Dr. Velez Melendez, and he will put up his PowerPoint and we will move on from there.
Well,
thank you for joining us.
Well, no, thank you so much for having me. Thank you for the warm introduction. I'm very happy to be here. Okay, and you can see my screen right now, correct?
Yes.
Perfect. Okay. So yes, so I will be talking today about some of the work that me and my students that we're doing in our lab at San Francisco State University. But first, how I got into all this. So for evolutionary biologists, and there we go. One of the big challenges or one of the big questions is to explain and to understand the factors that maintain this amazing biodiversity that we see, right? So a lot of people would be focused on the animals per se. So we see here 6 different species of frogs, 6 different species of birds. And since very early on in my career, in my studies, I was very interested in the diversity, trying to understand the diversity of the communication signals that they use, okay? And here, these plots that you see here, these pictures, are what we call spectrograms. And I'm going to take a couple of minutes to explain them because we're going to see several of those today. So the spectrogram is kind of like a photograph of the sound. And on the x-axis here, down here, what we have is time. On the y-axis here, what we have is the frequency or the pitch of the sound. So a sound with high frequency is a very high pitch sound. And the sound with very low frequency, it's a low pitch sound. And then in color, the brightness is how loud the sound is at those particular frequencies. So these are the communication signals of those animals that you just saw. So this is the great tree frog, very common in Minnesota where I did my PhD. And the call sounds something like this call. This here is our spring beeper. Very common also in Minnesota, sounds like beep, beep, beep. We have the Carolina chickadee with the Phoebe song, Phoebe. That's not their chickadee call, but their Phoebe song that they use during the breeding season. The nuthatches, no, not the nuthatches are these ones with a bang, 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 bang. And then here what we have are the titmice or the tufted titmouse with their Peter, Peter song, Peter, 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 Peter. So yeah, so this one I'm not even going to try to make the sound. But yeah, so these spectrograms are this representation of sound. And they're used, these are their communication signals, right? So, but what is communication? How do we even define communication? And at the most broader sense, communication is defined as an exchange of information from a signaler to a receiver in the form of a signal that passes through some medium. Okay, so we have here our signaler, our receiver, the signal that passes through some medium. And the type of information that can be encoded in the signal can be about the species identity. So here it is, so yeah, I'm a red-winged blackbird. Sex, I'm a male red-winged blackbird. My body size and fighting ability, physiological condition, genetic quality, a lot of different things can be encoded, or a lot of different types of information can be encoded in those signals. So if we look at all these, these type of information, we can start thinking, well, knowing what species the animal is, what sex, and then things like their fighting ability, their body size, their physiological condition, their genetic quality, all these are attributes that are very important in very important behaviors like attracting a mate, right? Or defending a territory, which are very related to at the end of the day, the fitness or whether some animals can pass their genes to the next generation. So because communication is so important to attract females or to attract mates, to defend their breeding territories, we would expect natural and sexual selection to favor these signals that can maximize or optimize the transmission of information from the sender to the receiver. And then in the receivers, we would expect sensory systems or mechanisms that analyze those signals that closely match those properties of the signals. So maybe many of you have heard about Darwin and his theory of natural selection, but I don't know if many of you have heard about sexual selection. But it turns out that Darwin also defined and proposed his theory of sexual selection. And one of Darwin's quotes that I really like is this. He's talking about sexual selection, and he says, this form of selection depends not on the struggle for existence in relation to other organic beings or to the external conditions, but on the struggle between the individuals of one sex, generally the males, for the possession of the other sex, generally the females. I like to highlight this here in red. He clearly explains what natural selection is, and here in blue, he compares it to what sexual selection is. So he also identified two major forms of sexual selection. One is male-male competition, so males competing to have access to females. And the other is female choice, females actively choosing which male to mate with. And in the cases in which females are choosing particular males, they tend to prefer things like this, very exaggerated traits. And that's how Darwin explained the evolution of these very exaggerated traits. So females tend to choose these exaggerated traits, whether they're louder calls, the longer feathers, or the longer songs, those that are produced faster. These exaggerated that seem to have a cost on the males to produce. So speaking of these feathers, one other thing that I also like, and how Darwin always thought about natural selection and his theory of natural selection and the problems that people could see. And one of the big problems was like this, right? Like, how can we explain the evolution of these peacock's tail by natural selection? And he even said, the sight of a feather in a peacock's tail, whenever I gaze at it, makes me sick, right? But as great as he is, he came up with in 1871, I would just say it because it's 150 years this year that we celebrate the publication of this second book, this big book on the descent of man and selection in relation to sex, where he explains all his theory of sexual selection. Okay, so but we're here to talk about frogs, not birds and their peacock's tails. So communication in frogs, or in frogs, the most common form or modality of communication is acoustic communication. They produce calls. We have, I think the last time I checked was over 7,000 species of frogs and toads, and almost all of them have their own species-specific distinct call, mating call. And again, here we have the gray tree frog, the green tree frog that sounds kind of like wah, 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 the spring peeper, the American toad, the boreal chorus frog, and then American bullfrog, so it's like round, round, round. I point these out because we have learned a lot of how frogs use these communication signals from some of these species. For example, From the American bullfrogs, we know that males defend territories. They will fight intruders. And we've learned that they know how to recognize who's their neighbor and who's an intruder. So they will not attack their neighbors. They know how to recognize them from the calls. They will only attack intruders. We know that one, or probably the most important thing to learn who's my neighbor and who's my neighbor is based on that pitch, on the frequency of the call, right? In these frogs, or in frogs in general, the frequency of the call is a good indicator of the size of the male. So think like in an orchestra, the violin, very small, very high pitch. Then you have the viola, bigger, lower pitch, and the cello, much bigger, much like a lower frequency or a lower pitch in the sound. So the same, the organs that produce the sounds, the vocal folds in frogs, the bigger the animal, the bigger those vocal folds, so the lower the pitch of the sound that they will produce. So yeah, so a lot of frogs, bullfrogs and other species, they can assess the size of the opponent based on the pitch of the call. Great tree frogs, we've learned from these great tree frogs that this call is kind of like a string of pulses. And females really like longer calls. And through a set of experiments, they show that actually it seems like females gain indirect benefits when they paired males or females with males that had either a short call or a male call. The tadpoles that were fertilized by sperm of longer calls, those tadpoles developed faster, got bigger faster. So it seems like there are some benefits that, some genetic benefits that the females will receive or their offspring will receive for choosing these types of males. And the last thing that I want to mention here that I always think it's very interesting. So these tree frogs, the great tree frog, the American toad, and the chorus frog, there are several nights during the breeding season that they will be breeding at the same time in the same ponds. And I recorded a lot of these frogs and I recorded a lot of the chorus noise, the noise. There are hundreds and hundreds of frogs, of males of each of these species calling at the same time. And it always is very interesting. We see the great tree frogs here. They have energy here like around 2.2 kilohertz and 1.1 kilohertz. The American toads are around 1.6 kilohertz, and then these chorus frogs are like 3 to 4 kilohertz. So when all of them are calling together, the great tree frogs are using like these two channels. The American toads fit like right here in between, right there. And then these boreal chorus frogs are calling up here, right? And when I looked at it, I was like, oh, it's like all of them are communicating in like their own private channel, right? So when you think about the signals in this case with these particular channels, we want those sensory systems to be very tuned to those channels. So we would expect that the boreal chorus frogs, their hearing is very sensitive up here at above 3 kilohertz or so, that the American toads may be very sensitive here around 1.6, and that the great tree frogs more sensitive at these two particular frequencies. And at least we know that for these guys, the great tree frogs, yes, they don't hear very well at 1.6. So it's kind of a way of filtering out those toads, the calls of those toads, and just paying attention to the sounds in their frequency range of their calls. Okay, so we have learned a lot about communication with frogs, but today I'm going to talk to you about our own Pacific chorus frogs, right? And the ribbit of our own Pacific chorus frog. And yeah, so 7,000 species of frogs, each of them with their own call, and really only the Pacific chorus frogs are the ones that sound ribbit, 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 right? So this is, again, the photo of their call. we define or we explain, they define this call as having two phases. So one phase here, each of these black things is like a pulse. So now in this one, we have time and then amplitude of the sound or how loud the sound is. So the sound is getting louder. And here we have, again, the spectrogram with the frequency. So here around 2.1, 2.2 kilohertz is where that is very strong, the sound. And then they have this gap. So this is the ret, and this is the bit, ret, bit, pretty much. Why did they become so famous? Well, they're very common in California. They're very common around Hollywood. That is the sound of the frog for every movie back in the day, right? When movies were starting to get big, well, we need a frog, record whatever it's out there. Even if it was a frog from South America, they will put the sound of the ribbit. And that's how that sound got that so popular. Okay, but it turns out that our frogs not only produce this type of call, they have two mating calls. And this is what they call the diphasic or two phases call. And they have diphasic and monophasic call. The other one is called the monophasic call. And here we have 13 seconds of a recording of one male. And I'm going to play, what you're going to hear is first 1, 2, 3, 4 diphasic calls, and then a series of monophasic calls. Let's see if you can hear this. Right. So what, like the first thing that we did when I started my lab here is go out and get a lot of recordings of these frogs. And an amazing student in my lab, Adriana Guajardo, she helped me a lot getting those recordings and analyzing and looking at the differences between these calls. So we described very thoroughly the diphasic and the monophasic calls. And what we see is that so the big difference is the diphasic calls have that silent gap, right? But these, as we can see here, they're delivered slower, right? The males take their time between one call and the other. The monophasic, they're produced much faster, right? These diphasic tend to be a little bit shorter, and the monophasic tend to be a little bit longer. And because they're longer, and they're produced at a faster rate, so they have like a high call effort. It takes a lot of energy, or we would predict that it will take a lot of effort, a lot of energy to produce these monophasic calls over those diphasic calls. So the questions now, and another student in my lab, she just started as a master's student, and she's very interested in finally deciphering why do we have these two different types of calls? Do they have different functions? Do females prefer one call over the other? And we'll predict that they would prefer the monophasic calls because they seem to be more energetically costly, more louder, faster, longer, like Darwin predicted. And then some of the things that we wanted to start looking into is what's going on in the brain when these females hear these calls. What is ticking in there that it's making them choose, hey, I like that call better than the other. And there are some neurons actually in their brain that at some point we'll like to start looking into. It's very interesting because these neurons count pulses. So think of this like those pulses of the call. They count, right? And they will only fire after there's, let's say, 10 pulses, right? They will not fire at 8. They will fire only until they hear those 10 pulses. And what's interesting is if you add a gap there in the middle, they reset and they start counting again, right? So those diphasic calls with that gap in the middle is a mystery, right? We would think, oh, and by the way, these neurons were discovered in our own Pacific forest frogs. So we know they have those neurons. So these neurons, we think, are going to be some of the neural substrates or the neural mechanisms behind why we see these different types of calls, and we can try to understand how females are choosing or analyzing these calls based on the responses of these neurons. Okay. One other thing that I mentioned a little bit was that match between the signals, right, and the receivers. And we were talking about how, yeah, we want this signal at this frequency, and to be a private channel, then the sensory system should be kind of like very tuned to those frequencies or to that channel. So now I'm going to show you yet another way to look at sound. And now it's like if we were to average or sum all these up, right? Like average throughout the entire call. So remember, we have frequency here on the y-axis. Now I'm going to flip it. I'm going to put it on the x-axis. And these color in yellow, I'm going to put it on the y-axis. So pretty much here, this band here, lower band here in red, it's this first peak here about 1 kilohertz. And this yellow band, more higher amplitude here, is this peak here at 2 kilohertz, right? So this, on average, when we look at the entire call, there's very loud sound at around 2 kilohertz, and then there's a second peak at around 1 kilohertz. And then this third one here, it's this bit here at around almost 3.5 or so. So the question is, here we have those frequencies. Is the auditory system tuned to the frequencies in that call? The way we do that, we answer that question, is using the same technique that we use with newborn babies to check their auditory system, their hearing, to see if they have normal hearing. And it's called the auditory brainstem response. And these auditory brainstem response just measures the neural activity that is caused by sounds, okay? So with that, we can play back sounds at certain frequencies and start asking, do you hear it or you don't hear it, right? But now with babies and with frogs, we cannot ask them, hey, do you hear it? No, we get the response from the auditory system. And here's pretty much what we get. So for example, this is a very loud tone, short tone. I think this was like 2 kilohertz or so, but this 85 decibels, it's pretty loud. So just for comparison, well, frogs call between 85 and 90 decibels at 1 meter. That is pretty loud. 90 dB is a truck like driving right in front of you, or a big motorcycle driving right in front of you. It's pretty loud. So what we get when we play back that tone is this trace. This is the response of the auditory system, okay? Now we can play back that same tone, but a little bit softer at 80 dB. We still get a strong response. We start lowering the volume of that tone. We see now that, hey, here you see there are several peaks when the tone is very loud. Here we start losing some of that activity, neural activity, and we only see this one. We see how that response starts getting weaker and weaker. Maybe here at 50 dB, we still can see something of the response. When we play that sound much softer or a little bit softer at 45 dB, we don't see any response, right? So we call like, okay, somewhere here between 45 and 50 dB, that's what we're gonna call the threshold, which is the minimum volume necessary to elicit that response, right? So now, if you think about this, lower is better, right? Lower means that you can hear that particular sound when it's very, very soft, very, very soft. So remember, lower is better. So we played these sounds from very low frequencies, 400 hertz up to 5 kilohertz. And here's what we find, right? So in our frogs, we see that they're very, very sensitive. They can hear very well these around 2 kilohertz. Around 1 kilohertz, they cannot hear very well. But then they have another area of high sensitivity. They're very sensitive around 400, 500 hertz. And this shape of the audiogram, that's what they call the audiogram, it's typically common in frogs, but it's very different from us. Like humans, of birds, if you look, it's more like a U shape, more like a U shape, one like very sensitive at mid frequencies and not so much at lower and higher frequencies. And that's because we have one inner ear, the cochlea. Frogs have two different organs. And then, so this one here, the amphibian papilla is tuned to lower frequencies. The basilar papilla is tuned to these higher frequencies. But okay, we were looking at whether there is a match. And it kind of is. There is kind of a match. This is the call in gray. Remember that figure that I showed you earlier? So this peak kind of like matches very well with that area of high sensitivity. This one, however, does not, right? And this is a little bit puzzling. And I still don't know how to explain very well this result. When you look at other frogs, typically this gray matches very well here, and this one matches very well there. So it's a little bit puzzling with our frogs. Okay. So that's where we're at now with looking at one of the populations in the frog. But going back to Darwin and his louder, longer, faster, right? And this idea that females are choosy, and if females are choosy and they really select for the louder, longer, faster, why do we still see males that produce short calls, soft calls, and very slow calls? How is that variation in the calls maintained? If females are so choosy, if females were so choosy, then we should only see males producing louder, longer, faster calls. Well, William Hamilton and Marlene Zuck, they proposed their Hamilton-Zuck hypotheses, and they suggest that health and resistance to parasites are indicators of good condition. And then that these preferred traits, the longer, louder, faster, are very costly to produce. And that only healthy males in good condition can produce these costly preferred traits. Okay, so that's how they proposed that's why we see this variation. Only those males that are resistant to parasitism or that are in very healthy and in good condition, those are the ones that can produce the preferred traits. So my student, Julia Messersmith, for her master's thesis, she decided that she wanted to test the Hamilton-Zuck hypothesis. And she studied the Pacific chorus frog and the fungus, I always cannot say the name correctly, so I'll just say BD. Is the chytrid fungus, is this fungus that it affects the skin of the frogs, and it has been killing frogs all around the world. Is one of the major causes, well, not one of the major, but it's one of the it is actually one of the major causes for extinction of a lot of species of frogs and toads. So our frogs, the Pacific chorus frogs, are actually very resistant to that fungus, and that is some line of research that is going on now at school. And it's why are they so resistant? What do they have that they can tolerate heavy loads or large loads of infection from those frogs? So Julia, for her master's thesis, she asked, okay, does BD infection affect male calling behavior, and whether females prefer calls of uninfected males? So some of her predictions based on Hamilton-Zuck. So Hamilton-Zuck say that these secondary sexual traits are honest signals of parasite resistance. So she predicted that she will find a correlation between the infection and some properties of the calls. Hamilton and Zuck proposed that more energy-dependent traits will be more affected. So because of this finding on monophasic and diphasic calls, Julia predicted that there will be stronger effects of BD on these monophasic calls than diphasic calls. And then Hamilton proposes or predicts that females prefer less infected males. So Julia predicted that females will prefer calls to the call of less infected males. So the first thing she did was go out and get recordings and get samples of the BD infection of the males. What she found was that all of the males that she recorded, 41 in total, all of the males were infected. Some of them with very, very high loads of infection, some with not so much, but all of them were infected. And here what I'm showing you is on the x-axis is how like a lot of fungus in them, a lot of very, very heavily infected by the fungus on the right, not so much on the left. And here the duration of the call, looking at longer calls, right? And what we see is that for both types of calls, there's this negative trend. The more infected they are, the shorter the calls that they produce. And the effect is stronger for those monophasic calls. I'm going to skip this one for now, but this is that trend, right? So if the male is heavily infected, it seems like it produces shorter calls. Well, this is not totally new. Several other papers have shown that in the past that there's some effect of infection by the fungus on their calls. All of these papers said, oh, this must have strong implications for sexual selection and mating behavior. None of those studies really actually test that, but Julia did. So Julia, we bring frogs to the lab, and in the lab we have this setup in which we have this anechoic, like this sound booth. It's very, very quiet in there. And we have a circular arena, and we bring a frog, we place it there in the middle, and we can play sounds from different speakers, and we can control what types of sounds we play back. And we can observe their behavior from a camera that we have overhead. And yeah, so Julia asked, hey, I'm going to give you a call typical of a heavily infected male versus a call of typical of a male that doesn't have much infection. And so this here is when we're watching from overhead. This is the speaker playing back the heavy infected call. This is the speaker playing back the low infection call. And here, I don't know if you can see, but right here in the middle, there's a cup, and here is our frog. There are two eyes there, and this will probably make more sense when we start it. So we're gonna start hearing some calls. You see some movement here. And there she goes, right? So this is one of the paradigms that we use to ask different types of questions about sexual selection, recognition, hearing in our frogs. So yeah, so these are very preliminary results, but we have tested 12 females so far. And of those females, 7 chose the low BD call, 5 chose the high BD call. So really, females don't seem to care that much. We're planning, as soon as the frogs start coming out, increasing our sample size here. But the big conclusion here is we cannot assume that differences in the signals may have implications for receiver behavior. Even though BD seems to be affecting the calls, our females don't seem to be caring that much about it. Okay. So another line of research in my lab is looking at anthropogenic or urban noise. And this is a topic that is very hot lately. A lot of people are very interested on how human-generated noise affects animal behavior in general. And this is one pretty cool paper that came out during the COVID-19 shutdown. The authors, Elizabeth Derryberry, she did a lot of recordings in the past, and she showed how white-crowned sparrows here in the Bay Area, in the city with anthropogenic noise, they have changed their call. And when COVID hit, she came back and she recorded the same males, and then she found how, well, if it's silent, we go back to call how we usually call. So it was a pretty cool study. And reading that paper, Tara, oh, well, yeah, so I'll tell you, one of my students got She was not in my lab. She read that paper and she said, oh, I want to do something like that. So then she transferred to my lab. And I'll talk a little bit about her data in a second. But anyways, that same author, Elizabeth Derryberry, they just published this what is known and not known about acoustic communication in urban soundscape. I'm going to tell you pretty quick what we know. What we know is how that city noise is. Again, here we have frequency, so high pitch on the right, low frequency, low pitch on the left, and how loud that noise is. And here in this first line, this is a forest, okay? Forest. And this black line, that's city noise. And what's important and what people show over and over again is that that city noise is very loud at lower frequencies, okay? So between 1 and 3 kilohertz is where that city noise is pretty high. And what we also know is how the signalers, the animals that produce the sound, are coping with that noise. So 3 ways in which they do it is if the noise is very loud, here in gray now it's a representation of this area here, right? In gray, we have the noise. This in blue is the signal of the, let's say the frog, for example, right? So what many people have shown is that, okay, when there's a lot of noise, animals, what are trying to do now is avoid those noisy areas. So they move to a less noisy area, or they produce their signals at times when the noise is low. The second strategy that they use is if the noise is there, they just sing loudly, right? Louder. They produce signals that are louder. So to increase that probability of hearing. Now here's the one that it's pretty cool. Because that noise is, the noise is stronger at lower frequencies, some animals, what they do is shift their signals to a higher frequency, right? To avoid that low frequency noise. So their calls or their songs are at higher pitch. So that's what we know. What we don't know, and that comes from this paper, the greatest gap in knowledge in the field is understanding how receivers may be adjusting or evolving in terms of their sensory capabilities and thresholds in noise, right? So we know a lot about how the signalers are changing their signals. We don't know how the receivers are changing. So that's what Tara joined my lab to work at. She started asking, how does natural and anthropogenic noise affect animal communication systems? Oh, here's Pacifica, right? So I think we have a pond there close to Pacifica. It's close to the ocean surf. We have a pond here in by the airport. We have a pond in the Presidio, and then in the GNRA and the Point Reyes, and some here in the East Bay. And today I'm going to show you data from these 2 populations, one in the Presidio of San Francisco and one in the East Bay by Briones. And this is a much quieter population. This is a much louder population close in the city. So the first thing she did was go out and get recordings from those frogs. And really, we don't see many differences. So again, the bigger the male, the lower the frequency, and that's the trend. But we don't see much of a difference between the urban population and the rural population. And in call duration, same. Not big difference, but again, we have very few recordings from the urban. What is interesting though, she started looking at their hearing, again, testing like the babies. And these are responses to a click. It's like those clicks that stimulate, very short, that stimulate all the auditory system. And what we're finding is that the responses of the auditory system are stronger to those clicks from frogs from the rural population. Now, when we look at the sensitivity, how good they hear the different frequencies, what we find is that frogs from the quieter rural population have lower thresholds, are more sensitive. Remember, lower is better at these lower frequencies compared to those frogs of the noisy urban population. And this is true for these, the region of this auditory inner ear organ, and also here all the way up to the higher frequencies. So remember, lower is better, higher is not so good. It's kind of hearing loss a little bit, right? So it's not to the extent that people would be concerned like, oh, that's hearing loss. Not really, but kind of, right? So they have this less sensitivity in at least this population. And again, please take this with a grain of salt. It's only 2 populations. This spring season that it's coming, we're trying to hit all those populations that I showed you in the map. And one of the ideas that we have is that those higher thresholds, actually, that instead of being hearing loss, could be an adaptation to hear better in noise. Let's filter out the noise, but not by not hearing it. Frogs still call very loudly, so it might be actually an adaptation to communicate in very noise populations. So Caroline, she's a new grad student in the lab, and she's gonna start tackling those questions. Can lower auditory sensitivity enhance or promote this communication in noise? And then she wanted to start looking also at what are the anatomical basis, start looking into the ear to see what's different between those populations. Why do some hear better than others? Okay, and the last piece that I want to talk about is how these communication in our Pacific chorus frogs can be related to the formation or the maintenance of different maybe species. There is great debate about how many species of tree frogs are there, or Pacific chorus frogs. Some authors propose that there are 3, Hyleola regilla here in the north, Hyleola sierra here in pretty much all California, and then more towards the east, and then Hyleola hypochondriaca in the south and down to Mexico. However, there are other authors that say, well, our results do not support that species designation, right? We do see these 3 different clades or 3 different lineages, but we don't see that there are 3 different species. And they propose further geographic sampling that incorporates behavioral data with multi-locus genetic data may be necessary to clarify the taxonomy. Anyways, I'm not a strong evolutionary biologist, taxonomist. I'm very interested in evolution, yes, but in the context of sexual selection, species recognition, and all these that I've been talking about, not in the taxonomy per se. So when I first moved to California, I was like, oh, this is perfect. I'm gonna travel all California. I'm gonna start recording the calls of frogs from all those different places and start looking, well, if the call is so important for reproduction, right? It's species specific, then we would expect to see differences in the calls between these 3 clades. So I started doing that, but then Javier Plasencia joined my lab as a graduate student, and he raised it up a notch. He said, no, I do want to look into the genetics, and I do want to look also not just the calls, but their morphology, right? So maybe he's asking, what is the role of ecological selection, sexual selection, and just pure random genetic drift? How do they interact to promote or maintain this separation between the groups, between these 3 different clades? So we've sampled different locations across California thus far, and I color-coded it kind of like from northern to southern in this green to bluish. And of course, I was saying these beautiful places that I've been to, like the field station over Sierra Nevada, and other of the field stations of the UC system, the University of California system. It's a great system. I've had so much time, such a great time going to those places to record. Anyways, what we find is that, so again, here's kind of like the trend. Dominant frequency, the pitch of the call tends to be higher in these blue populations, but doesn't seem to be that strong. However, I did some preliminary studies on their sensitivity from only these 2 populations, lower calls, lower frequency calls in this light green. And what we find is that light green, they're more sensitive at lower frequencies, which is kind of like what we expect if there's that match in their signals and sensory systems. So that's very promising on telling us that both the calls and the auditory system can change easily. But the big differences that we find is in the number of pulses, right? So these black things are the pulses. So here, the greener populations tend to have more pulses in this first part of the call, tend to have more pulses than the others. And now the strongest difference is in the rate, how fast those pulses are produced. So again, these greener populations have much higher rates than these lower populations. So yeah. So the big question there, and we know from other species that pulse rate, it might not sound as something very interesting and something like, wow, that's crazy to many of us, but for the frogs, it is. Like the frogs do pay attention a lot to that pulse rate and to that number of pulses. So the question is whether there's some behavioral isolation going on with the differences in those calls. And some of the questions that we want to ask now is, what are the implications for mating behavior? Do females prefer calls of their own lineage, their own population? How strong are those preferences? What call properties are used to recognize and prefer, if there are preferences, calls of their own local lineages? And then what are the neural bases? Remember those those pulses that count, right? They're very tuned to the number of pulses and to the pulse rate. So we predict that we're going to find differences in the tuning properties of those neurons. And then we can start looking, what are the genetic bases for those, for the tuning properties of those neurons. So I also showed how the periphery can be malleable and can change. So where do those preferences arise? So all of these are kind of like the types of questions that we want to address in the future. And with that, I hope I was able to convey a little bit about the all the research that we're going on in our lab on how these animal communication systems, and now I talk about animal communication system because it includes both the sender, the receiver, the type of the signal, right? How do they evolve? What are the factors that shape these signals and their hearing and their sensory mechanisms? And then with that, I would like to just thank all the members in my lab, Dr. Vance Redenburg, who helped a lot with the fungus part of that project. All these people from the different research stations that we went to around California. And then people here at the East Bay Mod to access to the ponds here in the East Bay, and then John Young and Natalie Reeder for their help and access to the ponds in the Presidio and the ponds by the San Francisco airport. And definitely thank you very much for listening. Thank you for having me. And I hope you enjoyed it as much as I did. I would be happy to take any questions if you have any, if there's time, I don't know.
Well, thank you, Alejandro. That was absolutely fascinating. Questions, please write your questions and put them in the Q&A section, press enter, and we'll have a look and see what you have to ask. It turns out that, of course, noise, as we've discovered even from whales and so on, from the studies on whales, that noise is very disruptive to them in terms of navigation and so on. So it would appear perhaps that, our noise that we create as human beings in various forms will inevitably have some negative effect on mating calls.
Yes, so definitely, like in frogs, actually, it has been a little bit understudied. Whales, there's been a lot of studies looking at, because those drilling underwater, that makes so much noise underwater. And there are studies showing like big impacts of that noise that we as humans generate on whales and marine animals in general. With frogs, actually, there was one paper with our own Pacific chorus frogs. It was conducted in Oregon, and they looked at some call properties with some populations close to the road and some others farther away from the road, and they didn't see that much changes in their calls, right? Same as we are. We're not finding that many changes or big changes in their calls. Yeah, and honestly, not many people, as in that paper in 2021, the big gap is we don't know what's happening with their ears. So hopefully we'll start to learn and adding to that part with Tara's and Carolyn's project.
So let's have a look. Bing Huey is asking, are the call properties constant over environmental conditions such as temperature or elevation?
Yes, no, so that's a great question. So frogs are like cold-blooded animals. So they're not. The rate at which they produce those pulses, the rate at which they produce the calls, they change with temperature. So typically, the warmer the water where they're breeding, the faster they call. And the faster they call makes the calls a little bit shorter too. So for all of these analyses we always, every time we record a frog, we take measurements of the temperature, and then we correct for that trend of temperature, or that dependence of those properties with temperature. So that is a great point. With elevation, so we need to look at that, because with elevation, there are so many things that change with elevation. So temperature can change, but also typically frogs from higher elevations tend to be bigger. And then bigger frogs, as we talked, they tend to have lower frequencies, right? So there is a suite of properties of both anatomical properties and properties of the call that can also change with elevation and environmental properties, right? So all of these, we try to control for all of these when we do our analyses as best as we can. Question.
And Deb Pierce is concerned about information being published about the negative effects of the noise produced by wind turbines. Oh, is this part of your research and or conclusions?
So yeah, so we are not close to any place with wind turbines, and there is a lot of research going on around those wind turbines. And actually, one of the lab where I did one of my postdocs at Purdue, they are very interested, and they were looking at eagles and different birds of prey, because they've been really harmed with the wind turbine. So we were trying to see how they hear, what they hear from them, and to try to see if there's some way, either visually or acoustically, that they can like move them away from there, right? So I think at this moment, there's a lot of research from different angles trying to see how can we mitigate the problems, right? How can we get other lines of research in those labs? We're like, okay, how do we get birds to stop crashing into airplanes? And they try to take a sensory approach. What is it of the bird that we can get them scared from the wind turbine, scared from the airplane? Yeah, so, and again, the wind turbines also have some effect on our hearing.
But the birds are actually flying on a particular pathway, which they've been flying forever more than a day. I mean, right. It's wired into them to fly a certain way. So they inevitably fly into these turbines. And plus there's this power force that's created by these turbines. I mean, there's a wind, wind currents that are just literally created by them that may even suck them in.
Yeah. Yeah.
So,
Yeah. Yeah.
but is there, with the noise element, is there a decrease in the population of Pacific tree frogs that has been
Of
discovered?
Pacific tree, I don't think so. We don't see these Pacific tree frogs are very common around the Bay Area, are very common pretty much around California and the West Coast. I haven't been here long enough to see any trends. I think the droughts are a bigger problem for them than the noise.
The which? The
Drought.
the
droughts.
The
Yeah,
droughts,
the droughts. The droughts and the changes to the landscape that can generate more droughts or get rid of their breeding sites, their ponds. That would be the main problem that I see with the frogs, more than the noise.
Uh-huh. So the heat and the fires and all of that are not bringing about any positive, obviously,
No.
results to.
No. Yeah.
In fact, just the opposite.
It's
And
the
Sharon
opposite,
is
yes.
asking, are we looking primarily at spectral content within our auditory range or also beyond, higher and lower?
So here with frogs is within our range. And anthropogenic noise, it's also within our range is about between 20 hertz and 20 kilohertz. It also depends. There's a lot of variation. My students were laughing at me yesterday because we were calibrating some speakers and I couldn't hear some of the higher tones. So yeah, but all of these, the frogs, the range that we're looking is between 400 hertz and 5 kilohertz. Well, a little bit lower. We're looking between 200 hertz and 5 kilohertz. All that is within our hearing range. Anthropogenic noise goes lower frequencies, but still within our hearing range because we hear down to 20 kilohertz. And then all the other stuff, it's more vibration and stuff. Yeah, and we don't look much higher than that. And I don't think anthropogenic noise gets that higher at higher frequencies. So bats, for example, in the ultrasound, they should not be that affected by human generated noise. They would probably be more affected by light and then by all the obstacles with all the echoes that they produce, probably.
Okay, so there's some, let's see what's happening in chat. So maybe there are.
Maybe I'll stop sharing here so that we can see my face bigger.
Thank you.
Sorry.
So with regards to chat, let's see. You can see this as well.
Yes, I just.
It says, do you know the health of the tree frog from populations in Pacific and around the Bay Area? Great talk, thanks.
Thank you. So again, with my very limited experience thus far, it has been the populations here are doing okay, I have to say. So the problem is I started 2018. 2018 and 2019 were great years with rains, great years with rain. 2020, well, no, 2019, sorry, 2019 was pretty dry. And it picked up towards the end. And then we saw a lot of frogs in these populations where we got rain, right? Then it was 2020 when COVID hit. And again, it was very dry until March or so. And right when it started raining, and right when I started seeing all the females in the field, we went to lockdown, so I couldn't go out there anymore. But I'm positive that last year with the rains that came after during the pandemic, they were doing okay. I'm very curious to see what's going to happen this year because it's been very dry. And when I look at La Nina forecasts and how the weather patterns are going to be here in the Bay Area, it's kind of like 50-50 chance it could be really good or really bad. So We'll just have to wait and see.
Well, when you're talking about rain, how much does, how do you calculate by inches, by the number
Yeah,
of
so
storms that drop whatever number?
so because I'm not that expert in rainfall and all that, I gauge it by, is there water in the ponds or not? And right now there's no water in those ponds. And by this time we should have water. And many of the ponds are still dry. So there has to be enough water that to fill out those ponds.
And the frogs during this period of time, they go into some form of estivation, hibernation.
Correct. They're just waiting. And with the cold weather, they kind of like, so our frogs, because here it doesn't get that cold, I don't think they go into a full hibernation really, but more like feeding, waiting, lower their metabolism during the winter, and just waiting. And the females producing those eggs and males accumulating that sperm and getting ready until the temperature rises a little bit and there's water in the pond so that they can lay their eggs and the tadpoles can develop. Yeah. Let's see what David asked above. David, okay. Are they competing when the chorus gets louder and louder? Yeah, and what does it take for one frog to decide to begin the call chorus? Okay, and when they, yeah, oh, this is awesome. Yeah, so I can tell that you guys hear those frogs out there because that's typical, that is a great description of how those choruses of these frogs go. Typically there's one guy that starts calling, right? And then the rest start getting like excited and calling, and suddenly it's a really loud chorus of a lot of frogs calling. And then all of a sudden, like they shut down. There's silence for a while. And then again, one guy starts calling, and so forth. So yes, there was a study back in the 70s. It was like at that time, it was like a big thing. It was one of the first studies with our Pacific chorus frogs showing how females show strong preferences for the leaders. And those leaders are the ones that start that chorus. In the study, I don't remember very well the details, but they run playback tests like the one I showed you. They had 4 speakers and they had one that was a leader and then the others were followers and females were choosing that. But then in the field, they also saw, they measured the size, the body condition. They observed, they waited during these bouts and they waited to see which one was the male that was starting the chorus. And what they found was that often it was the same males who started first. And then they measured and they found that those males tended to be in better condition than the other males, right? So it seems like depending on their condition is when they're more willing to start calling. And when they're more willing to start calling, they're going to attract more females. So calling is very costly energetically. So these frogs are calling really, really, really loud. And they're doing it like ribbit, 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 right? And they can hold that for minutes and that like they're going to be exhausted. So they have to be in really, really good condition to maintain this. So I think that condition, like the condition in which they are, is kind of like what decides which of the males start calling, right? And why they stop, it could be because either they're getting tired, they need to recover a little bit. Sometimes it's just a movement, right? Those calls are also attracting predators. So some movement in the water or some noise, typically like boom, they go down and they stay silent. And then once again, that male in really high conditions, like, forget it. I don't care if there's another predator there. I just want to mate and start calling. Then all the others start like, okay, it's safe. So let's start calling back again, right? So it's kind of like that. If you're calling by yourself, the probabilities that a predator will get you are higher. Once one is calling, okay, now it's 50-50 chance if I start calling with him, and then the other, well, maybe now it's a third if I start calling with them, and then they start, like, the cores start getting big. Are they competing? Yes. Yes, so I didn't show it, but they also have another type of call, is the aggressive call. And when another male gets too close to them, they don't like it. And they want to, they are not territorial in the sense that they defend the territory, but they do have their personal
space. Yeah.
right? They have their personal space that they don't like to be to get other males close by. So when another male calls very close by, they switch to this aggressive call to try to keep each male separated. Now, what's pretty cool is in nights when there are very few males calling, that personal space is bigger. When there are a lot of males calling, like they have to tolerate getting closer, right? It's like getting into BART, and if it's empty, you don't want someone to just stand by you, right? But if it's packed with a lot of people, then you tolerate people like standing by, right? So it's pretty much like the same thing. We think also that monophasic call often, now that you listen to those frogs, it's often the case that they start with the diphasic calls, ribbit, ribbit, ribbit. And when more and more males start joining the chorus, they get more excited and they feel like there's more competition. And typically that's when they switch to the monophasic call. And that's why we think maybe that monophasic call is more expensive to produce and it might be more attractive to females. But if it is, then why do they do the diphasic? Maybe to not spend too much energy if they don't need to.
So there seems to be quite a few questions still that need to be addressed.
Oh, yes, there are a lot of questions to answer here.
And with the ones who call first, are they sort of like the alpha males, the ones who?
Yes, kind of. So from what they showed from that study in the 70s, not really the alpha male because they don't have like a social group and hierarchy.
Right.
More like those who are in better condition, right? Those that are well fed and have a lot of energy tend to be the ones that start the chorus. Yeah.
Well, you always need the ones who have the initiative.
Yes.
And then the others join in, and then you have this wonderful sound out there that just, just gorgeous to listen to, and the females must love it.
Yes,
it's
they do.
Yeah, right. Okay. Let's see, what's it?
See what else we have.
Anything?
I don't see you.
What David Edel asked above, please.
Oh,
I think we.
Yeah, so that one was that one, I think.
Yeah.
Let me know if there's anything else. I see. How do they ID the frogs in the Q&A? And by that you mean us as researchers, or the different frogs? How do they identify other frogs? So in our frogs, there have been some studies on like habituation, but it's not really, well, a little bit. Well, not in our frogs, if I remember correctly, but there's some like, yes, like some males will habituate. If there's another male calling very close by, at first they will get very aggressive, right? And they will start calling aggressively. But the problem is that the aggressive calls, they don't attract females, right? So the male has to really weigh in, okay, how much do I want to fight and how much do I want to attract a female? So at some point, that habituation, the male becomes tolerant to that other male and start producing again the mating call and not the aggressive call. And what's cool is as soon as you make a little change on that call that was making him first aggressive, but now he habituated, if you change that call, sometimes the male will detect that, oh, this is another guy, right? And they will be aggressive again. And that's how pretty much how the bullfrogs learn their neighbors. They to the type of noise, they learn that type of noise and or call. And if there's a change in that call, then it's like, oh, that's not my neighbor, right? So that's kind of like how they identify the different males in the population. But because our frogs are territorial, it's not like they're really learning who their neighbor is. It's more like, hey, you're too close or you're not that close.
Yeah. With the low, I don't know whether, well, there's one question that I have with the lower sounds that the males make and the females seemingly to like the lower sounds that the males make. It sort of reminds me of singers and,
yes.
and the fact that the lower voices tend to be very sort of more
attractive, dare I say?
More the women, the females or the women are more attracted to the male singers that have lower voices. What can I say? You know, like the baritones and not necessarily the bass, but you know, there's a range. And I'm thinking these chorus, the females are not crazy. They know what they like. You know,
they know what they like. Yes.
Yeah, they know what they like. Can you blame them? Exactly. No, at least you know when I'm listening, it's like.
So Deb says, to be clear, please, the monophasic is like a legato sound heard in music. I'm not that familiar with the term, but yeah, it's more like a longer like instead of the ribbit. I don't know if that's
like legato in music is when the notes are connected to one another. There's perfect. A continuity. Yes.
Okay, so yes, so kind of, because it's like that gap is filled with more notes. So right. There's more of a continuity. Yeah, that's great. Thank you. I'll use that in my future seminars. Okay, so when the frogs get going in the pond below where we live, the rabbits are indistinguishable and and remind us of a legato sound. Yeah. So yeah, no, when all of them are there, it's just it's really, really hard to tell. It's really hard to tell the different frogs. For us, it's very difficult. So that was kind of like some of the work that I did during my PhD and the lab where I did my PhD. That was one of their big questions. Like you've heard those frogs in your ponds and you see that there are a lot of frogs, like really, literally, there are hundreds of frogs in very small ponds. How does a female go in there and, hey, I like that one, right? It's kind of like, it's related to, in humans, we call it the cocktail party problem. And it's when there are a lot of people talking, music, a lot of noise, a lot of voices. How do we piece apart, like which voice comes from which person? Because in the ear, all it comes is a combined sound and our brain has to break it apart. Oh, that's the guitar. Oh, that's the drums. Oh, that's the conversation I'm holding, right? So how do frogs do it in those very, very noisy situations? Yeah.
Any explanation? No.
No, not yet.
Not yet. And another question that has to be resolved sometime in the future.
Yes.
Well, it looks like you've answered everybody's questions. And let's see. This is a very musical group we have. It says, do they harmonize? Do they actually harmonize?
No.
No,
they don't.
This is not a four-part chorus or choir.
Yeah. No, unfortunately, they don't. It's more very like a cacophony out there. It's
yeah.
Yes.
Cacophony would be a good sound. Yeah, good word. Yeah.
They don't do a barbershop quartet.
No, I think we have to go somewhere else for a barbershop. Yeah, barbershop quartet. Yeah, I think so. It's very nice, but I don't think the frogs are into that somehow. It's not their
I wish they were.
But
I wouldn't be losing my hearing that much if they were.
Well, everyone says thank you for your time and expertise and for sharing it. And I, on behalf of everyone present and everyone who was part of all of this, just want to thank you so much for.
Oh, thank you, thank
for
you, really, thank you
fascinating.
so much for having me.
It's the first time we've had anyone present on Pacific chorus frogs and
There
the
we
whole
go.
concept of vocalization and communication. And
There we go.
it's just thrilling to have someone
Yeah.
who's a specialist in it.
So now when you hear them in the next few months, now you'll think more about what those males are trying to do. And
And we'll pray for more rain.
thank
So-
you. Thank
yes.
you. Yes.
We
yes.
definitely will. We
Oh,
need.
yes. And when you start hearing them there, please drop me an email. I'll be
We'll do.
check them out.
Okay.
Keep an
Absolutely.
ear out there for us.
All right, folks, if you hear any after the rains, let me know and I'll email Alejandro and let him know and you give me the location and he'll be right there.
Awesome.
So we've got something to keep us busy as soon as the rains come.
There we go.
Excellent.
Perfect.
So again,
Perfect.
thank you so much.
Thank you. Thank you so much.
Good night.
Bye. Good night.
Good night.
Good night, everyone.
Ooh, I have heard one or two already. I know they're coming.
One question. Sometimes
Yes.
I wonder, am I hearing the chorus frogs or crickets? And I probably would at night it would be the frogs, right?
Most likely, yes. The crickets will come later in the summer. Yeah, but yeah, most likely the frogs. At first, like at this time, sometimes you'll hear some of them during the day and they'll go like, and those are the poor guys that are, they want to get there now. But it's too early, but yes. You want to be the first one in the bar. So
Yeah, right. The first one through the door is the one who has the drink, right? So
There
it's
you
like,
go.
hey, yeah, man, right. Okay.
Yeah.
So
Okay.
anyway, we'll end this
Okay,
on a
yeah.
very positive note, and we're going to just have a terrific evening after this and think of
Awesome.
all the wonderful things you've told us.
Perfect. Thank you so much. I really appreciate it. Thank you.
Thank you so
Okay,
much.
thank
Good
you.
night.
Night.
Bye.
Okay, so whoever is left, I'm going to end the webinar at this point. So it will be, it has been recorded and will be available in a week or two on the website. So please stay tuned if you wish to hear it again. By all means, go to our website and you can listen to it again and enjoy all the wonderful information that Dr. Alejandro was able to convey to us. Thank you again for joining and all the very best. Take care.